continuing in chapter 22, we are still focusing on the metabolism, also you could think of movement, incorporation of nitrogen into molecules in the body. But now we want to focus our attention to nucleotides. So just a couple of review slides. What do we need to make nucleotides for? What is their purpose? Well, we don't produce nucleotides as a way to store energy. Nucleotides are not going to be degraded for energy production. Nucleotides are instead going to be degraded if there's an issue, but not for the purpose of producing energy or anything like that. And now listed on this slide, just remember, why are we making these nucleotides? Nucleotides are, of course, polymerized to produce our DNA and our RNA. Coenzymes that we've talked about the entire trimester, NADH, FAD, coenzyme A, and the list just goes on and on of the functions of nucleotides. To remind you, you need to make sure you're good on the terminology associated with nucleotides, as well as the basic structure of nucleotides. Now, I'm not expecting you to draw and manipulate these nucleotides, but if we're in the process of understanding how we are building a nucleotide, it would be helpful if you remember the structure of a nucleotide. So make sure you go back and review the different classes of nucleotides. We have purines, smaller word, bigger molecule. These are our double rings, adenine and guanine. We have our pyrimidines, found in DNA, thymine and cytosine, found in RNA, cytosine, and uracil. The terms here, this is the term for the nitrogenous base. When the nitrogenous base is combined with a sugar, that sugar will be either ribose or deoxyribose. The nitrogenous base plus a sugar forms a nucleoside, and each nucleoside has a name. This is the nucleoside with a ribose and the nitrogenous base cytosine. This is called cytidine. This is the nucleoside with adenine for the nitrogenous base and deoxyribose for the sugar, forming deoxyadenosine. You have to also recall that if we have a nucleoside, we can add phosphates to make a nucleotide. You have the option of adding one phosphate to this nucleoside, 2 phosphates, or 3 phosphates. So you can have a mono, a di, or a triphosphate of any nucleotide. And last bit of information to go back and review is the structures of the less common nitrogenous bases. So we have xanthine, and if we make one little change, we produce hypoxanthine. These also can form into nucleosides by adding a sugar. So just make sure you go back and look at these structures and this terminology. So as we go through the biosynthetic pathways, we're going to look at both Denovo pathways and salvage pathways. We can produce purines and pyrimidines in both ways. With our pyrimidines, the most important pathway will be the Denovo pathway, meaning completely from scratch. With our purines, our body's going to more heavily rely on the salvage pathway, which means we're going to take some portion of a nucleotide and just fix it to use that to build back to the nucleotide that we're trying to produce. Alright, so starting at the beginning, we have our dietary nucleotides. So just like most of our molecules we've studied, we're going to ingest nucleotides. They can be broken down in our digestive tract, and some will just directly be eliminated. Others will be broken down, and then they can enter into a salvage pathway, travel through the body, and be incorporated into DNA, RNA, or other molecules made from nucleotides. It is important to remember we're not using these for fuel. It's not efficient for us to use nucleotides in this manner. So let's start with the most complex. Let's build a purine molecule de novo. So we're going to build adenine and guanine from scratch. The best way to learn this is to begin by studying this image, where you can look at the important molecules we use to produce our purines. So starting here, we see formate is going to provide some carbon as well as bicarbonate. Glycine provides a couple carbons and a nitrogen. The entire molecule is going to incorporate. And then we're going to obtain nitrogen from glutamine and aspartate. To produce a purine, we first need to produce a molecule called phosphoribosyl pyrophosphate. This is called PRPP. To produce PRPP, we take ribose 5-phosphate, made in the pentose phosphate pathway, and we add 2 phosphate molecules from the donor ATP. The enzyme that accomplishes the production of PRPP is termed PRPP synthetase. PRPP synthetase is activated by inorganic phosphate, but it's also going to experience a feed-forward activation, where the more substrate you have, the more products you produce. We're going to inhibit this reaction by the presence of purines. We're going to see this repeated over and over again in nitrogen metabolism. The ultimate product is going to come back and feedback inhibit some of the preliminary reactions to prevent excessive production of something that we already have. All right, so we have our PRPP. We're ready to build our purines. With purine biosynthesis de novo, PRPP is going to be our starting product, our starting block. We're then going to build the nitrogenous base on the sugar, producing an intermediate of inosine monophosphate. And from there, we'll produce our AMP and GMP. So let's take a look, starting with our PRPP that we just produced. We're going to incorporate a nitrogen by the donor glutamine using the enzyme glutamine PRPP aminotransferase to produce our first product that is exclusive to purine biosynthesis. The glutamine PRPP aminotransferase is the committed step of this reaction. Now that we've incorporated this nitrogen, this molecule is destined to become a purine. So we continue through the steps, adding a glycine, then a carbon by way of this formate derivative, then a nitrogen again. A little bit of ATP allows us to close our ring. We continue. Bicarbonate gives us another carbon. Aspartate donates really almost an entire molecule, but then we'll have some leaves, so we end up with aspartate just giving us the nitrogen. Then another folate derivative comes in, gives us a carbon. And finally, we close the ring, forming this common intermediate called inosine monophosphate, this IMP. Now with IMP, we can branch out and produce either AMP or, following the bottom pathway here, GMP. We're producing AMP and GMP, our purines, in both the monophosphate form. We can then come back and further phosphorylate those molecules to produce ADP, ATP, as well as GDP and GTP. One thing to note here in this branch point, notice we're going to alternately use energy sources. So for AMP production, we don't break down ATP to produce AMP. That would be counterintuitive. So we use GTP as our energy source. Always fall back to your knowledge that we need fairly equivalent amounts of these nucleotides, because when we build DNA and RNA, we've got those complementary base pairs. And so you can see at the bottom, to produce our GMP, we use an energy source of ATP. Regulation of adenine and guanine biosynthesis is fairly straightforward. we're going to have our products, AMP and GMP, going back and inhibiting some of the key enzymatic steps in the production of AMP and GMP. So make sure you take a look at this regulation. Now let's take a look at the salvage pathways to produce purine. As I stated initially, this is really important for the production of purines. Notice in this image, we can take free bases, so these are our free nitrogenous bases, and they can all have a addition of PRPP to produce a nucleotide. So this is in this middle column here, we've got a sugar and a phosphate attached. We can also take the nucleotide, remove phosphates, and end up with our nucleosides. We can take nucleosides, convert back to a nucleotide at times. We can do that with adenosine. With adenosine, we can also deaminate, producing inosine. With inosine and guanosine, we can remove the sugar, going back to the free base. So these may not look like very complex or even ultimately very important reactions, but there are a few disorders that I think are worth you knowing that really demonstrate the importance of the reactions. If we lose the ability to convert adenosine into inosine, this one deamination reaction catalyzed by adenosine deaminase, this can lead to a disorder known as severe combined immunodeficiency, where a patient will have absolutely depleted levels of T and B cells. This can lead to death or a very, very heavily medically intrusive treatment simply because of the loss of the activity of one enzyme. We also can see a very intense disorder by the loss of the action of this enzyme, hypoxanthine guanine phosphoribosyl transferase, this HGPRT. If we lose the function of HGPRT, the disorder is known as Lehninger Syndrome. It's a very devastating neurological disorder. Proves how heavily the brain relies on these purine salvage pathways. And then lastly, if we lose the function of this purine nucleoside phosphorylase here and here, we end up with a PNP deficiency that also has a huge impact on our immune system cells, specifically the T cells. Now there are times when we end up with purines that we need to dispose of, and when our bodies are capable of breaking down purine. But remember, this is not to produce energy. When purines are broken down, we see more of a convergence now, where the AMP first loses a phosphate, then is deaminated to inosine, we lose the sugar to form hypoxanthine and ultimately xanthine. With GMP, similarly, we lose the phosphate, we lose the sugar, and then guanine is deaminated to produce xanthine. Xanthine will then be converted to uric acid by this important enzyme, xanthine oxidase. This is a very straightforward breakdown process, but we see some pretty severe issues if we have any type of error in the breakdown. This can be excessive breakdown by an overactive xanthine oxidase, or simply just we have too much uric acid produced because we have too many nucleotides for some other reason. When we have excessive amounts of uric acid, we call this disorder gout. There's lots of causes for gout, so I listed just a few. Some of it can be a renal issue where we're not really making too much uric acid, we're just not excreting it properly, so this is a kidney issue. We can see the overproduction of uric acid because we have too much PRPP being made. High amounts of PRPP will lead to an increased production of purines, so we have more around, we need to break them down. Gout is going to cause some pretty, I hate to use the word interesting clinical features, but we can see swollen joints because we actually will have build up crystals of uric acid. We see deposits in the ear. This is a pretty bad example. It can cause types of arthritis because it can get into our joints and our fingers and toes. We can even see large levels of buildup of these crystals that will cause us to need surgical removal of the crystals.